Okay, on the previous slide, we were looking at bacterial pathogens, a phylogenetic tree of pathogens. And so there are, of course, bacteria that cause disease, but what are those diseases and how do they cause those diseases and the symptoms that go along with the disease? And this is a very short list, as I'm sure you realize. But pathogens produce these symptoms by either locally or what we call systemically. So locally means where the bacteria is infecting you, that place in your body, the cells and tissues are being damaged. So at the site of infection. In other words, if you get a cut and that cut gets infected, that pathogen is infecting your skin at that site of infection, at that damaged area, but it's not really affecting your whole body. It's not, the bacterial cells themselves are not traveling throughout your body. Their toxins might be traveling throughout your body, but for the most part, the infection is localized just to your skin, for example. Systemic is body-wide, by either the cells themselves getting into your bloodstream and traveling throughout your body, or their toxins getting into your bloodstream and traveling throughout your body. And these toxins interfere with your normal metabolism. And there are two types of toxins. Exotoxins, which kind of goes along with that term exocytosis, if you'll recall from our last unit. This is how cells secrete things outside themselves. And prokaryotes can do this too. So exotoxins are compounds that are secreted by living pathogens, basically as their waste products. You know, they're just getting things out of the cell that they don't want in the cell. And so they're not trying to produce toxins, and they're not trying to make us sick. Their waste products simply are toxic to our systems, to our cells, and to our metabolism. Then, whenever you see this exo, there's usually an endo counterpart to it, and there are endotoxins, which are actually cell components, like pieces of membrane and proteins from the bacteria that are released when the bacteria dies and lyses, breaks open. So when the cell disintegrates, basically, it releases compounds into your bloodstream that act as toxins, that interfere with your metabolism and interfere with cell function. So when it comes to these diseases, Lyme disease is basically systemic. It starts off in the skin, but the toxins, at least the toxins, if not the organisms themselves, travel throughout your body and cause the infection. Tetanus, same thing. The infection starts out localized, but the organisms themselves and definitely their toxins will travel throughout your blood and your body and cause these symptoms. Tuberculosis is in your lungs. It's an infection of your lungs, so you've got a bacterial colony growing in your lungs, and of course their toxins are traveling throughout your body. So even though this is localized to your lungs, the toxins are definitely going systemic, and that's why you end up with body-wide symptoms like this. Same with bacterial meningitis. Meningitis is caused by there's viral meningitis too. So the meninges is the covering of the brain. So this is any infection of the covering of the brain is meningitis, the meninges. So that's the localization of the infection is the meninges. But then again, the toxins travel throughout your body and cause all these different symptoms. And then strep throat, same thing. Of course, strep throat is in your throat, but you feel terrible all over because the toxins are entering your bloodstream and going systemic. So all of these have both localized and systemic effects to make you sick. And one thing I should point out in preparation for the upcoming test, we're studying viruses and bacteria here together, so you need to be aware of which diseases are viral and which diseases are bacterial. We looked at a table very similar to this one for viruses. So go back to that table and make sure that you know which diseases are caused by which pathogen. Okay, the description below here relates to this figure. So I'm just going to read through this with you and then point out where we are in the figure. in relation to it. But what we're looking at here is vaccination and how vaccination works. And vaccination works because you have an immune system. So you'll understand how vaccination works better if you have a better understanding of how your immune system works. And this is just like a crash course in your immune system. But a vaccine is made from an antigen. And an antigen, here in the diagram, is any piece of a pathogen, or it could be the whole pathogen itself, but it's been modified so it can't make you sick. But it could be just a molecule, a compound, a protein from the pathogen, a piece of the pathogen. But that's what we refer to as an antigen. An antigen is anything that's going to cause your immune system to react, cause your immune system to mount an immune response, as we say. So this is a piece of the pathogen or the pathogen that's been changed so it can't make you sick. The vaccine is injected into the bloodstream. The antigen is injected into the bloodstream. We learned on the previous slide that there are two parts to the immune system, innate and acquired. The innate immune system pretty much begins with your macrophages. Once a pathogen gets inside you, once an antigen gets inside you, the macrophages are patrolling your body. They're like little amoebas that crawl around inside you and look for foreign invading organisms or pieces of organisms. And in this case, again, antigens could be just molecules that shouldn't be in your body, pieces of organisms. They, as there is shown here in the diagram, the macrophages engulf the antigen by phagocytosis, if you'll recall that from our last unit. It's a form of endocytosis. And the macrophage then will present the antigen to help to T cells, and these can be either killer T cells or helper T cells. On this side of the diagram, we're looking at killer T cells, but we also have helper T cells in the diagram over here. But the killer T cells are called killer because they will, for example, in a viral infection, they will seek out and destroy the cells that have been infected by the virus. So they kill your cells. And that's where the symptoms come from. It's really not the antigen that's killing your cells necessarily, or the pathogen that's killing your cells necessarily, but your own immune system is killing your cells because they've been infected. So that's what the killer T cells are doing. B cells in the bloodstream, with the help of helper T cells, so that's what we see going on over here. This is a helper T cell that's kind of activating the B cell that has picked up the antigen. The B cells then pump out antibodies, little proteins that glom onto the antigen or the pathogen and prevent it from infecting cells. That also signals macrophages to eat the pathogen. So if this is a bacterial pathogen that's infecting you, the antibodies are going to glom onto that bacteria, and then macrophages will come along and engulf the bacteria, just like we're seeing here with the antigen. So this neutralizes the antigen itself, or the pathogen itself. And then once the pathogen has been eliminated, no longer is making you sick, there are memory cells, both T and B memory cells, that are produced that remain ready to mount a quick adaptive immune response against repeat infection by the same pathogen. So that's how you're that's the part of your immune system that remembers and learns what the pathogen looks like. So if the same antigen, the same pathogen gets into your body again, it will immediately jump on it, and B cells will almost immediately start pumping out antibodies. So the body doesn't have to learn, the immune system doesn't have to learn all over again what the antigen is and what antibodies to produce. The B cells already know, they remember, and they start pumping out antibody against that pathogen right away, and you don't get sick. It keeps you from getting sick. So with that background, you should be able to understand how a vaccine works. And we have vaccines for bacterial pathogens just like we have them for viral pathogens. So, for example, you can get a vaccine against pneumonia. Pneumonia is caused by bacteria growing in your lungs. Actually, I need to get one myself. So, like I said, a vaccine is a piece of a pathogen or the whole pathogen itself that has been inactivated, so it can't make you sick. And what it does is it trains your immune system to remember that pathogen, and that provides immunity. And what immunity is the ability to resist a particular pathogen or toxin by the action of the immune system, and that would be the acquired, the part of the immune system we refer to as the acquired immunity. So innate prevents initial infection. Your skin is your primary barrier to being infected by pathogens. Your nose, you're breathing in pathogens all the time, but your nose has hairs and mucus and mechanisms to prevent your infection. So hairs and mucus and enzymes, like in your tears, there are enzymes that will actually cause the lysis of bacteria that get into your eyes. Macrophages are patrolling and eating invaders, pathogens. Killer T cells are going around destroying cells that have been infected. So that's all part of your innate immune system. But then we have the adaptive or acquired immune system, and that's the part that is involved in vaccination. That's what makes vaccination work. It learns and remembers how to fight off a specific pathogen. Specific. So you probably have heard you never get the same cold twice. Your immune system has learned, your adaptive or acquired immune system has learned what a cold looks like. So once you get over that cold, you're immune to that cold. The next cold that you catch is a different a variation of that cold virus. That's the only way it can make you sick again. Otherwise, you wouldn't get sick again with a cold after you've been sick with a cold once. So it's because the cold virus changes so quickly, being an RNA virus, if you'll recall. So the part of your immune system that is adaptive, macrophages learn dendritic cells, which are similar to macrophages. They pick up antigens and they present them to T cells and B cells. They go looking for the right T cells and B cells when they pick up antigens. And then helper T cells activate B cells, help to activate B cells, and then the B cells pump out antibodies. So that's how vaccines work, but of course we also have antibiotics. And the word itself, antibiotic, means against life, or something that will kill something. Bio meaning life, right? So in this, like if you go to the doctor and you're sick, and the doctor cultures the organism that's making you sick. So for example, you have a sore throat. The doctor will swab your throat and inoculate a petri dish with agar in it and grow the bacteria, and then apply different little discs, paper discs. These are made of paper, and each of these little discs is soaked with a different antibiotic. And in this way, the doctor can tell which antibiotic is most effective at killing the bacteria that's making you sick. So this is a way of learning the sensitivity of the bacteria to different antibiotics. And the flip side of sensitivity is resistance. So this also will show which antibiotic is being resisted by the bacteria. In other words, the bacteria is resistant to that antibiotic. And we look for this thing called the zone of inhibition, which is around each disc here, except for this one, because this one must be the control. This is probably soaked with just distilled water, so there's no antibiotic on that disc. That's why there's no zone of inhibition around it. So that's the control disc, and these other ones have various different antibiotics. This one, for example, has novabiasin. That's this one here, and you can see that novabiasin doesn't have a very big zone of inhibition around it. This one's similar, but the zone of inhibition around this one looks a lot clearer than the zone of inhibition around the novabiasin disc. So it looks like novabiasin is the worst antibiotic in this case. It's the one that the bacteria is most resistant to. And if we zoom in on that one, and that's what we're looking at here, all these little bumps within the zone of inhibition are bacterial colonies that are still able to survive. This bacteria has evolved antibiotic resistance and is able to grow even in the presence of that antibiotic. So the bigger the zone of inhibition, the better the antibiotic works against that bacteria. The least resistant the bacteria is to that antibiotic, or you can also say the more sensitive that bacteria is to that antibiotic. So an antibiotic is a medication. It's used to kill prokaryotes, living pathogens. That's why it doesn't work against viruses, because viruses are not living. You can't kill something that's not alive. So we only have vaccines against viruses. The disc with no zone of inhibition is the control, likely has distilled water. Antibiotic resistance refers to the capacity of pathogens to not be killed by antibiotics. The ability of a pathogen to resist being killed by an antibiotic, like the novobiacin here. Bacteria may become resistant through mutation or horizontal gene transfer. Remember, conjugation, for example, a copy of the plasmid will be transferred from one bacterial cell to another, and that plasmid might have the genes for antibiotic resistance. So in that way, a bacterial cell can give another bacterial cell the ability to resist antibiotics, and that antibiotic resistance can spread through a whole population of bacteria that might be making you sick. Again, the opposite is antibiotic sensitivity, which means the degree to which a pathogen can be killed by an antibiotic. In other words, how sensitive is it to that antibiotic? How good does that antibiotic kill the bacteria? And in this scenario, these ones with the huge zones of inhibition, the largest zones of inhibition, show that this bacteria is sensitive to those antibiotics. And so those are the antibiotics that the doctor would want to give you to get rid of that pathogen, to get rid of that bacterial infection. So the zone of inhibition is the area around the disc where bacterial growth is inhibited. So the bigger the zone, the better. The better the antibiotic is in killing that bacteria. And bacterial populations most likely to be sensitive to an antibiotic during reproduction. In other words, most antibiotics function to prevent bacteria from successfully undergoing binary fission, and a lot of them target the cell wall. So in other words, the bacteria is not able to build cell wall when it is reproducing, when it's undergoing binary fission, and that prevents the bacteria from the bacterial colony from growing and making you sick. So based on these results, as I already pointed out, the antibiotic that should be most effective are the ones with the widest and clearest zone of inhibition. So it's not just the widest, but it's also the clearest. Again, for example, these two have about the same size zones of inhibition. They're both relatively ineffective, but the novobiacin is really ineffective because of these little colonies that are growing within the zone of inhibition. So that's how this system works, and you need to know this. I've seen this on the end of course exam, so it's an important concept to have. Not only because of that test, but this is all we have to fight off bacterial infection. And as you have probably heard, there are superbugs out there that have evolved antibiotic resistance to all the antibiotics that we have, so we can't kill them. And if you're infected by one of these antibiotic-resistant bugs, antibiotic-resistant bacterial strains or species, you can very easily die from that infection. So the best thing is prevention, and there are a number of ways that we prevent being infected by bacteria. And I'm sure you're well aware of every one of these, but prokaryotes, bacteria, pathogenic ones are the ones we're most worried about, can be controlled by refrigeration. So refrigerators, keeping food cold, will prevent bacterial growth on that food because it slows their metabolism. You know, they're not warm-blooded. They slow down. If you cool them down, they slow down. Everything about them slows down. So their entire metabolism slows down. They reproduce slower, and that prevents bacteria from growing on our food. That's why we refrigerate our food. Disinfection, Lysol, for example, and I chose Lysol because it has the word lice in it. The reason it's called Lysol is because it causes bacteria to explode. It causes them to lyse. That's basically what lysis is. Cells opening up, cells breaking open, exploding. Cooking, thorough cooking. So we want to bring hamburgers, for example, to 160 degrees Fahrenheit. Doesn't matter if it's still pink inside, as long as the meat on the on the inside of the burger gets to 160 degrees, that will kill any of the pathogens that might be in the burger. Like the E. coli from the cow that was butchered to get the meat from. That's the E. coli that will make you sick. You already have E. coli in your digestive system. You've got your E. coli, and that E. coli is helping you digest your food. Really, it's helping you prepare your non-digestible waste in your large intestine. But if you get E. coli from a cow in you, that's a different strain. That's a strain that's supposed to be in a cow, and it might make you sick. It might start multiplying inside you and making you sick. And then finally, we have sterilization. So heat and pressure kills most pathogenic prokaryotes. And so a pressure cooker will do this, and that's how we can things. For example, if you're going to can, I don't know, preserves, any kind of fruits or vegetables, you put them into the jar. You put the jars into a pressure cooker, and that pressure cooker is able to raise the temperature to much higher than the boiling point of water. And the contents, though, that are in the jar are not going to boil because it's under pressure. So the pressure keeps the water in whatever it is that you're pressure cooking from boiling, and that allows the temperature to go much higher and therefore kill anything that might be in those preserves or vegetables that you're preserving or canning. An autoclave is used by professionals, microbiologists, and hospitals, to sterilize equipment, sterilize scalpels and the instruments they use for operations. But it does the same thing as a pressure cooker. It's pressure and heat that allows the heat to go much higher than you would be able to apply otherwise. That would cause everything, all these liquids, all these solutions here to boil away. So that's the prevention. And you should practice prevention whenever you have the opportunity to. Oh, and one more thing that's not on the slide. Prevention includes trying to protect yourselves from being bitten by ticks, for example, so you don't get Lyme disease, being stabbed by a rusty nail so you don't get tetanus, and keeping up with your tetanus vaccination. So that's all part of vaccination in general, is all part of the prevention that is important here.